Thank you for inviting me to come and talk to you guys about bioinformatics today. Complete change of pace and topic to what Natasha was talking about. How many of you guys here know anything about biology? Some? Yay. How many of you did biology like in lower school, in high school? Yeah, that's me too. Everything that I talk about in terms of biology, I've learnt on the job. So please don't think that you can't get into bioinformatics if you didn't study biology. There are actually some quite well-known bioinformaticians who started out having done their PhDs in astrophysics or other areas of physics and have now gone on and are full professors in bioinformatics at leading medical research institutes. So if this is interesting to you, then please feel free to come and talk to me. But in the meantime, we're going to talk biology, and where was I? Oh, there it is. So the central dogma of biology, as it's known, is that we have DNA, and I hope that everyone is aware of your DNA. That's your sequence of A's, C's, G's and T's. In every cell in your body, you have the same complement of DNA, and you received half of your DNA from your mum and half of your DNA from your dad. That DNA content of every cell is the same, but obviously your cells are different. Your hair cells and your skin cells and your eye cells are different. They're doing different things. And that is because this sequence, or this DNA, is being transcribed into messenger RNA, which is then translated into proteins, and these proteins are the workhorses, and they're the things that go off and do things and make the differences and make your skin cells different to your nail cells. Right? It's not because the DNA content is different, but it's because different proteins have been turned on. And so at each level of your cell, we have this regulatory process that's going on somehow that is deciding what proteins are going to be active, turned on, produced in your cell, and which aren't. Okay? That's our basic cell biology 101 as told to you by a non-biologist. So hopefully there's not too many mistakes. What I do as a bioinformatician is I might be looking at someone's DNA. And we have whole genome sequencing. So we have beautiful biotech machines that will sequence the genome of people in about a day. Costs about $1,000. Produces a lot of data. There are 3 billion base pairs in the human genome. And what we're doing, partly at the DNA level, is we're trying to identify mutations, genes, signals that might be related to disease. So we take these 3 billion base pairs from lots and lots of individual people, and we end up creating pretty graphs. Biologists love pretty graphs, where for every single point along the genome where I have changes occurring, I'm looking at, this is the log base 10 of the p-value. So a higher log score is an extremely small p-value. And I'm looking for associations between that chromosomal region, that DNA locus, and a particular phenotype of interest. So actually, if my phenotype is continuous, like here I've got white matter hyperintensity lesions. It's a volumetric measure coming from MRI of people's brains. And I am trying to associate those volumetric measures, which are continuous of course, with different genetic genotypes, loci along the chromosome. And I'm basically running lots and lots and lots of regression analyses where my Y variable is my phenotype and my X variable is going to be a factor which represents the genotype at that particular locus. And then I end up with this lovely visualization which tells me that if there are associations, genetic associations, and particularly what regions of the chromosomes they might be, which region of the chromosome, which chromosome they might be occurring in. And then obviously I would then want to go and find map and look to see is there actually, because correlation is not causation, these are just correlations. I then have to go and find the causative mutation or the causative gene that might be impacting on the differences that we're seeing in our disease phenotypes. So that's one of the things that we're doing when we're looking at the DNA level. We're also looking to see copy number changes. You have 2 copies of each chromosome, which means you should have 2 copies of each genetic region. But sometimes, especially in cancer, things happen and regions are deleted or duplicated, and that might also impact on the phenotypes. And again, we're just looking for these patterns, and we're looking for the patterns of association that are related to diseases and other physical characteristics that we might be interested in. So let's go back, and now let's look at the next level, which is our transcriptome transcription, this messenger RNA. And there are 2 main levels that we can look at this. We can look at this originally in terms of DNA microarray techniques, which would give us an image, and each of these dots would represent a probe related to a gene, and we're looking to see whether or not there are differences, say, between the transcriptome of a normal patient versus a cancer patient. And we're looking to find differential expression. Now we've got RNA-seq, where we're looking at the transcriptome, and we're sequencing it the same way as we're now sequencing the genome. And we're looking for differences between normals and diseased, or 6-month-olds versus 20-year-olds versus 60-year-olds, right? Always we're looking at either a developmental process or we're looking at some sort of disease process most of the time. There has to be a reason for doing these experiments. Again, now we can get RNA-seq from a person. I think it's also less than $1,000. So it's pretty good. And what are we using this for? Well, this was the first example. This is the Dutch Cancer Institute. Back in 2001, had a landmark paper in Nature where they used the DNA microarray technique, because RNA-seq was not yet available. And they had 78 patients, and they had their tumour samples. These were breast cancer patients, and they were then able to use a basic classifier to find 70 genes, and on the basis of their gene expression, or the levels of these 70 genes that were present as measured on the microarray, they created a prognostic signature to whether or not people were at high risk of dying from breast cancer or whether or not they had a low risk signature. And obviously then we're going into personalised medicine. We're tailoring our medicine. If you've got a low risk signature, then maybe you don't need to have those aggressive chemotherapies. But if you've got a high risk signature, then maybe we're going to throw a lot more things at you. And so we're starting to use, and that now there are many, many examples of this, where we're measuring people's genetic, transcriptomic, and also proteomic data, and we're using that to try and diagnose who's going to get a disease, who's going to have a condition sooner, before they start to have the physical characteristics of that disease. And we're also starting to use biomarkers, these sorts of signatures, to try and dictate what sort of treatment people should be having, and using that also to guide. So, as I said, that's the DNA and the transcriptome, but then we also have the proteome. And the proteome is often measured using mass spectrometry. And again, there's a lot of biology, technical stuff in here, but at the end we get data. And we're generating a lot of data now. We're starting to be able to do proteomics genome-wide. We now have, of course, the Australian Phenome Centre based at Murdoch, which is doing large-scale mass spectrometry on people, metabolomics, phenomics. We like our omics technologies in bioinformatics, all generating more and more data. And then we're looking again at finding the patterns. So we're looking, these are the steps that we might be looking at in terms of if we've got proteins, we need to match the peptide spectrums, assign the peptides to proteins, and then we can do all the same sorts of things as what we did with the gene expression data and the genetic data. We're looking for the patterns, we're looking for the associations, the differences between our normal people and our diseased people. So that, you think, is everything. But wait, there's more. The genome is, our cells are extremely, extremely complicated. So we've always viewed DNA, messenger RNA, and proteins. Our view of DNA is that it's a straight line, right? It's this big, long sequence. But it's not. Our DNA is tightly packed. In our cells, we have chromosomes. And these chromosomes are very tightly packed chromatin structures that are wound around nucleosomes before we get to the actual DNA letters, strands that we see. And maybe you've seen DNA as being a double helix, but this double helix is itself tightly compacted and has other structures that it's wound around the nucleosomes. So then above the genome, we have the epigenome. And we can measure different aspects of the epigenome now, also on a genome-wide scale, at a fine scale, using sequencing technologies. So these nucleosomes have these histones, are made up these 4 histone proteins, and we can have modifications on these histones, and we can measure them. We have DNA methylation, which is the addition of this methyl group onto our cytosines. Because somehow our cell has to tell its DNA somehow what gene it wants to transcribe. And that gene, if you like, a little bit of machinery has to come down onto that DNA and read along it. So it's got to be accessible. And so the cell can control what genes are accessible by controlling how tightly packed the chromatin structure is by using chemical signals that will allow the chromatin to be relaxed or open. We can also measure using high C. We can measure what parts of the chromosome are touching each other when they're tightly wrapped or in certain states of the cell. And that also can tell us what's going on. We can also look at, I hope, that each cell in your body is replicating in mitosis. And we can look at, there might be changes in when different parts of the chromosome are replicated, depending on whether you've got cancer or normal. And we can do that, we can look at the replication timing of different genes and see if there are differences between different conditions or different cells in your body. So this is an old picture now from Nature, where we now have the Epi Genome Roadmap, where we have measured using standard techniques. There are 127 reference epigenomes from different cells in the body, and we've got genome-wide measurements for all of the marks from the chromatin states, from RNA-seq, DNA's accessibility, and whole genome bisulfite sequencing, which is methylation. So we have this huge cube of data, and this is just reference genomes. We are now obviously also generating this, well, not quite so fine scale, but we're generating it in a lot of the cohorts. For instance, I work with longitudinal cohorts looking at healthy aging. Why do some people age well and some people end up with dementia? So we've followed healthy cohorts over the last 10 years. We are starting to get whole genome sequencing data on these guys. We also have some methylation data on them. We have a little bit of RNA-seq data on them. And we're looking to try and find the patterns. What's causing some people to end up with dementia and other people not? What's causing some people to live longer, happier and healthier? And why do others get into problems? So that's a lot of data. But wait, there's more. Because we can now measure at the single cell resolution. A lot of what we've been talking about, or what I've been talking about, has been measured. For instance, we've been using, if I go back, so here I took a tumour, a tumour biopsy from the patient, and they put it on the microarray. But that tumour consists of hundreds of cells, thousands of cells. We know that there's a lot of heterogeneity, that not each cell in each tumour is going to have the same mutations, is going to be from the same origin, and is going to have the same characteristics. So now we can take a tumour and we can look at individual cells in that tumour. So now instead of just having 78 patients, one sample for each patient, we might have 78 patients and we'll have 2,000 or 3,000 cells from each patient's tumour and 2,000 or 3,000 cells from the normal adjacent tissue. So again, we're scaling up. And we now won't just measure the transcriptome, but we'll also measure some aspect of the epigenome at the same time. So our scale of data is getting up. We're not quite at Natasha's stage yet, but we're getting there. I mean, we just had 500 genomes, whole genome sequenced in Canada, and we had to have 5 hard drives shipped to us. So one of our issues is data and data storage. You want your compute to be next to your data storage, and we're also investing heavily and using Pawsey and other supercomputers and cloud computing a lot. There's a project in the UK called the UK Biobank where they have 500,000 people aged between 45 and 65, and they've done a lot of these screenings, if you like, of each of these 500,000 people, and this month they are going to be releasing the exome, which is not the whole genome, it's just the coding regions of the genome, sequenced for 300,000 people. And that's going to be available in the cloud for us to use to access. So I can't end without an acknowledgement of all the people that I work with. The great thing about bioinformatics is that we work a lot with people all over the world in large international collaborations also. They have cool logos and acronyms.